In this lecture, we're going to be looking at the contents of the cell, all the stuff that we find inside the cell, including the cytoplasm, and we'll get look at the first set of organelles. We'll save the rest of the organelles for a second lecture. So what do we find if you were to peel back the cell membrane and take a look at what's inside? Well, we've got four things that we'd find inside of a cell. There's the cytosol, Cytosol, think here now, cyto meaning cell, sol meaning solution. It's mostly water. It's about 90% water. And there's a very high concentration of proteins, so 10%-ish or so is mostly proteins. And that includes a whole lot of enzymes that are dissolved in the cytosol. And with all those dissolved proteins in there, it has a gelatin-like consistency. In case you didn't know, that's what gelatin is. It's a protein that we dissolve into warm water, and then we add sugar and color to it. And it's those dissolved proteins that kind of capture water molecules in a big old web and hold it in that, jiggly kind of shape. And so that is very similar to what the contents of a cell is like. It's got a lot of those dissolved proteins. You can find also inside of the cell what are called inclusions, and these are going to be crystallized chemicals or things like a lipid droplet. So it could be a big old oil droplet, and that's what we find inside of an adipose or a fat cell. It could be these granules of glycogen, which is that storage product of glucose. It could be pigments that are all kind of granulated in there. It could be a water vacuole, like a little bag that holds water, a bag of membranous bag that holds some water in there. So inclusions don't generally have, they're not doing any kind of, they're not performing a function. They have a function, there's value to them there, but they're not there doing work for the cell. They're just storage of some material. There's the cytoskeleton, and if we think about our bones as being the human body skeleton, the cytoskeleton is a supportive structure of proteins that are all that are wrapped together to form a rod or to form tubules, and they create this supportive framework inside the cell. And it's almost like they can be railroad tracks. So they act like railroad tracks and provide a pathway for moving things through the cell. And then last of all, in the cytoplasm, we find the organelles. These are the metabolic machinery of a cell. So they're busy performing the functions, the work that a cell has to do. So that's where we're headed next, then, is to look at what are some of the different types of organelles we find in a cell. So we'll start with the ribosomes. Ribosomes have a job of assembling proteins. They take information from the DNA, and so the DNA will send out from the nucleus a copy of the genetic code in the form of RNA, and that acts as the blueprint, the instructions that the cell needs in order to build the protein that it's looking for. A ribosome is composed of two parts. There's a protein part, and then there's another part that's composed of a different kind of RNA called ribosomal RNA. So this is ribosomal RNA is specifically there with the function of helping to produce the proteins. And these ribosomes may be found free in the cytosol, just kind of out there. And if that's the case, then when the RNA is delivered to the ribosome to be read and converted into a protein, then the protein is released into the cytosol, and it's there for the cell to use. But frequently, the ribosomes are found attached to the surface of a structure called the Endoplasmic Reticulum. We'll talk about that in a minute. And if that's where they're found, then the proteins are packaged and moved. So the proteins are placed inside the chambers of the endoplasmic reticulum, and then they can be packaged up and either moved into the cytosol in a package, or released from the cell, taken directly to the cell membrane, and then delivered to the external surface of the cell. So they can be in two locations, and the locations determine where the products are going. If it's a ribosome that's just floating free in the cytosol, then the protein that it makes is just going to be there, dissolved, one of the dissolved proteins or part of the cytoskeleton there in the cytosol. But if it's delivered, if the ribosome is attached to the endoplasmic reticulum, then that protein is delivered into the chambers of the ER and then packaged up to be delivered around the cell or delivered outside the cell. So if the ribosomes are the assembly workers, then the endoplasmic reticulum acts a little bit like the assembly line, where whatever products are being made are built and then can be delivered around the cell. So the endoplasmic reticulum, endo meaning inside, plasmic meaning talking about the fact that it is a system of plasma membrane, and then retic means network. So this is a network of plasma membrane found inside the cell, is what that name actually directly means. And it's this network of sacs and canals and little structures called vesicles. Those are just little packages that the endoplasmic reticulum can bud off, carrying material throughout the cell. The ER, the endoplasmic reticulum, often abbreviated as ER, that ER is connected back to the nuclear membrane. And that makes sense because there are, those are the instructions, that's where the instructions for making the proteins are found. And so it gets it's really closely associated with the endoplasmic reticulum. And the membrane of the endoplasmic reticulum, whether it has ribosomes on it or not, that membrane is going to be embedded with a lot of enzymes that can help to make chemical reactions happen more quickly. And that's what we mean when we say catalyze enzymatic reactions. That catalyzing process reduces the amount of energy required to do a chemical reaction. And because that less energy is required, it's easier to get the chemical reaction started. And so those enzymes are found there. They make chemical reactions happen, and they can make it happen very rapidly. And we use those chemical reactions to build molecules and to break down molecules, all that are associated with with the cell. So if the endoplasmic reticulum has ribosomes attached to it, then we call it rough endoplasmic reticulum. And it has this really, if you look at this electron micrograph here, you can see the little balls of the endoplasmic reticulum attached there. And it does give it a really kind of rough, studded appearance. And it's the rough ER that's found closer to the nucleus, because the ribosomes that are found on the endoplasmic reticulum are going to be building the proteins, and the nucleus has all the instructions, has the DNA, the instructions for how to build those proteins. So bringing them close together makes a lot of good sense. So as the proteins are built, so they're being built, then they would be placed in between the membranes, and then more enzymes can get to work on it, and eventually little bits will bud off. So if we pinched off that, if you can see where I'm pointing, so here, I'll try it here. If we pinch off that little corner of the endoplasmic reticulum, and there's some proteins that have been built inside there, that can be then turned into a vesicle, a little sac of membrane that has material in it, and then we can move that material to the Golgi apparatus, where more work can be done. We can do more chemical reactions that will change the protein to get it closer and closer to the finished product that the cell is trying to make. Further away from the nucleus, we see a transition where the endoplasmic reticulum becomes, where the ribosomes are no longer found attached to the endoplasmic reticulum. So here's an example or a picture there. And without ribosomes, then of course it can't make proteins. But we still find enzymes embedded on the surface of the membrane. So remember, it's a phospholipid bilayer, and there will be enzymes here and maybe enzymes here on both sides of the membrane that can make chemical reactions happen. Some of those are about building lipids with smooth endoplasmic reticulum, and of course the dietary fats, cholesterol, and the steroid hormones that we make from cholesterol, as well as synthesizing the phospholipids. I should probably have that in there too. Phospholipids. So we can make any kind of lipid depending on the cell. That will determine exactly which kind. But the endoplasmic reticulum would be the place where we would start building them. And it also, the smooth ER, has a role in detoxifying things that are toxic. So let's say we drink a bunch of alcohol, then the smooth ER that is very abundant in liver cells will go to work on that toxic alcohol, breaking it down into non-toxic side products for us. It also can, this smooth ER will detoxify pharmaceuticals that we take. There's a lot of chemicals that get produced in the body that we introduce into the body that, if they built up, would have a bad effect upon the body. And so we can count on our smooth ER to help break up and destroy those toxic materials. Okay, so we said that after the endoplasmic reticulum has made these materials, then they get packaged up and sent off to another location in the cell. And one of the other locations we can send it to is the Golgi apparatus. So I think of it as either the finishing and the mailroom, the place where we package things up. So the Golgi apparatus are these stacks of flattened membrane-bound sacs. And you can see them here. Here are the sacs. There's one here, another one, there's another one right there in this electron micrograph. And they're flattened, and they can be fairly large across. It's almost like a plate full of pancakes is what it always looks like to me. And it's here at the Golgi apparatus that we are able to, after we pinch off, so if this is a bit of endoplasmic reticulum, so the plasma membrane will kind of infold. So this is ER. Let's just keep this straight. It'll infold, and we can add more and more membrane here until eventually it pinches off. And so the ER now has a new nub, and this piece that just pinched off is now on its own. It can be moved throughout the cytoplasm. And what we move it to, it travels, is to the Golgi apparatus. So here's the Golgi apparatus. And so that little vesicle will come, it'll bump up against the Golgi apparatus, fuse with it, so now that wall is no longer there. And the material that was inside there, so again we're pinching that off because it was full of stuff, the final product, that material now is inside this first pancake of the Golgi apparatus. So there are specialized enzymes there that will cause chemical reactions to happen, and by the time it's done processing, then it'll pinch off, so now we have a new vesicle, and the product is further refined, and it can move from one layer to the next, and as it moves, it is further and further modified until we get to the end, and it pinches off this bit here, and that bit goes away with the final product of whatever it is that the cell was making. So if this is a cell in a mammary gland and we had a lactating mother, then the products that would be found inside that vesicle would be milk. So it would be full of milk proteins, it would be full of milk sugars, and it would be full of the lipids, the milk fats. And so all of that would then be waiting until baby cries again and says it's time to eat, in which case all of those vesicles would rush to the surface and release their contents. Okay, so the Golgi apparatus is all about taking the proteins, the lipids, and other materials that get synthesized or made at the endoplasmic reticulum, modifying them, doing the finish work to make sure that they are exactly what the cell was intending to produce, and then packaging that up into a vesicle so that it can be delivered to the cell surface or maintained. It could be maintained inside the cytoplasm of the cell. It can also produce new plasma membrane. So as I said, the endoplasmic reticulum can make phospholipids. And so the membrane that gets produced here, that membrane may just be bumped up against the cell surface. And then whatever was decorating the inside of the vesicle, oops, sorry about that. Whatever was decorating the inside of the vesicle is now just a part of the cell surface itself. So if this is the cell surface, that was a terrible picture. I don't have any space to draw, but hopefully you get what I'm saying. And if you don't, please ask me some questions. Read some more in the book. So the transport vessels that I've been talking about are the vesicles. And these are small membrane-bound sacs, and they contain a material. So here we see a vesicle way up close. This is not from a human body. The only image I could find was from this insect science website. But we see a membrane around it that is built of a bilayer of plasma membrane, just like the cell membrane is. And it's used to transport materials through the cell, to bring materials into the cell, and to remove materials from the cell. And these form at the cell membrane as small amounts of fluid are brought into the cell. So this is the cell membrane. Then a little pit can form here, and that pit will eventually close off. So these will grow as we add more phospholipid, and when it pinches off, it makes a little vesicle then that is released into the cell. Let me try that again. So here we have the cell surface, little indentation, and then cell membrane forms. That indentation gets further defined until eventually it's all but enclosed whatever material was outside the cell. So here's the material we're trying to collect and bring in. So we pinch it off here, and now it is free to travel throughout the cell, carrying whatever we brought it in. So that's one way that we can make a vesicle. But we can also make vesicles as the endoplasmic reticulum and the Golgi apparatus, bud off vesicles that contain whatever product that, whatever stage of the product that they are in production of right at that moment. So as we make materials at the ER and the Golgi apparatus, then vesicles are formed. So now let's try to put all this together to see how things are produced and transported in the cell. So, and this is, oh gosh, I went the wrong way. There we are. Here's my example of a milk-producing cell, a mammary gland cell. So here at the smooth endoplasmic reticulum, lipids are produced. And so we get vesicles filled with lipid being produced by the smooth endoplasmic reticulum. Here inside the nucleus of the cell, we recognize, it's time to make some milk protein. So a quantity of messenger RNA, a kind of RNA that can carry, because the DNA is not allowed to leave the cell, the messenger RNA can leave the cell. So we make a copy of the DNA onto messenger RNA. And I don't need you to know these kinds of details. But the messenger RNA is allowed to leave. It goes to find itself a ribosome. So here are some ribosomes. So the messenger RNA can sidle up to it, kind of lock into it, and we can assemble the protein using all of the information contained in that genetic code from the RNA. We also make sugars at the smooth endoplasmic reticulum and in the Golgi apparatus. And sometimes they get attached to proteins, sometimes they're just secreted out into a little sugar pouch inside the cell, made out of membrane that was a part of the Golgi apparatus or smooth endoplasmic reticulum. That material is just wrapped up in bits of Golgi apparatus or endoplasmic reticulum. So the proteins that were getting made up here, so we had proteins that were being produced thanks to the mRNA. They also get secreted into vesicles up here. They're transported down to the Golgi apparatus here. That's the purple stuff, is the Golgi apparatus. So those recently made proteins made by the ribosomes at the rough endoplasmic reticulum, those proteins are packaged up, delivered to the Golgi apparatus, and modified until we make the correct kind and amount of lactose so that the mother can feed her baby. So all of the milk proteins are vesicled up, all of the milk fats are vesicled up, and all the sugars are also vesicled up and secreted in. And so when baby is ready to nurse, she'll cry. Oxytocin gets released. That's a hormone. Don't have to worry about that. But that causes the rapid release of all of these little carbohydrate balls or vesicles, vesicles with the sugar in it, vesicles with the milk fat in it, and vesicles with the protein. All of them get released here at the cell surface, and ta-da, we got food delivered to the baby who is nursing. Okay, so those are all of the membrane-bounded organelles that are required to make and transport materials around the cell, with the exception of the ribosome, which has not got a membrane around it. So the ribosome has not got a membrane, but endoplasmic reticulum, Golgi apparatus, and vesicles are all membrane-bounded organelles. That means they have a membrane wrapping around it. Okay, as always, any questions? Email, office hours. Talk to you soon.